Alright, this video is a pre-lab video for your biomolecule lab. What you'll be doing is biochemical testing. The four main biomolecules: proteins, carbohydrates, lipids, and nucleic acids. In this lab we're going to be testing for proteins, and we're going to be testing for two different types of carbohydrates, one sugars and starches. So we're going to be doing proteins, sugars, starches. So one thing I do want to mention, the test that we're using for sugars is used to identify the presence of a specific type of sugar called reducing sugars. Now, what a reducing sugar is not necessarily important for our class, but what is important is the type of reducing sugar that we're identifying, and that is going to be glucose. So the sugar that we're looking for is going to be glucose, and so the tube needs to be labeled with G. Okay? The starch is going to be labeled with an S, and the proteins with a P. So we'll clearly be able to see our positive controls. When we use the protein, that will be egg white protein, so egg albumin. And then the starches, we're going to be using some potato starches. Okay? So the test that we're going to do for protein is called a Biuret test. And that test consists of two solutions that have been mixed together. One solution is going to be some copper sulfate, so copper 2 sulfate. And it's going to also contain some sodium hydroxide. Sodium hydroxide is what we call a strong base, and it does have some fairly corrosive properties, so we want to be very careful, and we'll be goggled up for this lab. For your sugars test or your glucose test, we're going to be using a Benedict's test. Now, Benedict's test has only one solution. Keep in mind the copper sulfate and the sodium hydroxide are mixed together, so we have collectively they're known as a Biuret solution. And then our Benedict's test is going to be conducted with a Benedict's solution. Okay. The iodine test, or what we use to test for starches, is going to be a combination of two different elements. One is iodine, or I should say iodine element, and some potassium iodide. Now, what happens when we mix these things together, we get something called IKI, and you could also use grams iodine for this, but we're going to be using IKI for our starch test, or to test for our starches. We're going to be using Benedict's solution to test for our sugars, specifically glucose. And we're going to be using our Biuret solution, which consists of copper sulfate and sodium hydroxide, to test for our proteins. Now, how our lab is going to look, or what we're going to be conducting, we're going to be doing some positive controls and some negative controls, and then you're going to be testing an unknown. So you're going to have two separate experiments going on at once. First, you're going to do your controls, then you're going to do your experimental. So my controls, which contain the egg protein, so my protein test, you're going to do the Biuret test of two things: the egg protein, which is labeled P, and water, which is labeled with a W. Now, why are we testing water? Well, we know that water doesn't have protein, so water is going to act like our negative control. Okay? Egg whites, which we know has protein, is going to act as our positive control. Now, the other two, same thing. We have our glucose, right? That's going to be our Benedict's test. And our two solutions, we're going to have a glucose solution, which obviously is going to test positive for glucose, and that will be our positive control. And then we also have water, which we know water does not contain glucose, so it should act as our negative control. Now, one thing about the Benedict's test is we're going to be adding heat to this. So there'll either be a water bath or a Bunsen burner, something that will allow you to add heat once you've added the Benedict's solution. Okay? So the Biuret test for the proteins, that's just going to be adding the solutions, and then you'll see a color change. The Benedict's test, you have to actually add the solutions and then apply heat. Okay? And in class, I'll tell you which heat application we're going to use. Lastly, we have our starch test. And this one is like our burette test where, so iodine test. This will just be a simple, once I add the solution, once I add the IKI to the samples, we get a color change or not get a color change depending on what we're looking at. So our positive control, or the solution is going to act as our positive control, is going to be some potato starch, because we know that potatoes contain starches. Okay? And then, again, like the other two, water is going to act as our negative control because water does not contain starch. Okay? So those are our three tests. The amounts and the exact procedure that we're going to do in class, I will talk about before we get there. This is just a background video, so you know what we're testing, how we're testing it, and what our positive controls are going to look like. Now this is the first collection of data that you're going to have. The second is I'm going to give you two solutions. I'm going to give you solution A and I'm going to give you solution B. And solution A and B both contain either one or more of the egg starches, or egg, potato, or glucose solutions. Right? So what you're going to do is, it's unknown. So you're going to conduct all three tests for A and then all three tests for B to identify which biomolecules are present in solution A and which biomolecules are in solution B. So you're going to have three different tests for A, and then you're going to do the same three tests for B. So you'll do, okay, didn't spell burette right. Let's try that again. Still spelled wrong, but yeah. Burette test for our protein. Spelled right there. That'll be testing for our protein, so we'll test for protein in solutions A and B using the burette test, just like you have done with your egg and water. And then you'll have your Benedict's test for testing for glucose. Now, the Benedict's test can test for other sugars other than glucose, but for this class, we're just going to be using it to test for glucose. And then lastly, we have our, not IKI, but our iodine test to test for starches in both solutions A and B. All right? So, grand total, you're going to be doing, for P, one test here, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So you're doing a grand total of 12 total tests in this experiment. Okay? 6 of them are to determine what your positive and negative controls look like, and 6 are going to be to identify any biomolecules present in your unknown.